Okay, so where we left off yesterday was talking about this thing. Let's remove this and try again. So when we have an experiment to find out the rate of photosynthesis of like an aquatic plant, right? When they ask us to describe preliminary practical work, we want to talk about like the suitable ranges, the suitable methods of gathering data, of setting the experiment, what at what temperatures, for how long, stuff like that. So we could do suitable methods of collecting the dependent variable, which um, in this case is collecting volume of O2 produced. Methods, I mean, suitable. Yep, suitable um, methods of changing the independent variable. In this case is I think this is good enough. Now over here when we talk about the actual detailed method. So let's look at this very carefully. There's 9 marks, and over here there's labeled 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 marks that you can score. Okay? Most of these should be pretty easy. Clear statement of the dependent variable. In this case, it's exactly the same as over here, where we mentioned, hey, what's the first we want to figure out the method of collecting the, in this case that is the dependent variable, how we can change the dependent variable over here, sorry, independent variable, and then over here how do we change the control, like what is going to be the suitable control variables. So you can think of this as always looking at, hey, like what is the independent variable of this experiment, what's the dependent variable of this experiment, what are the control variables, and finding suitable methods and suitable ranges for those variables is basically that preliminary practical work that they're talking about. Whereas over here, when we want to figure out a detailed method, it's asking you to include how you would control and monitor important variables. So not only how the experiment's set up and the experiment apparatus that you need, but also how you would control and monitor the important dependent, independent, and control variables, how you would control those things. Okay, so in this case, we already know we're going to be having a plant inside a water beaker. There's going to be foil on one side, a light filter on the other side, and a lamp. And we're going to be putting sodium hydrogen carbonate, and we're going to be measuring the volume of gas uptake with a photosinthometer. And that's pretty much the entire experiment. We're going to be changing light intensity by moving the light away and further and closer towards the beaker, and we're going to be having measuring that entire distance by using a ruler. Okay, so okay, first you want to get a clear statement on what the dependent variable is. In this case, it's obviously going to be the volume of gas that's going to be released. That's how we're going to be measuring the rate of photosynthesis. So the rate of photosynthesis is going to be determined by the dependent variable, which is going to be the amount of O2 that's produced. Description of the method of, so here's the dependent variable. Here's how we're going to measure the dependent variable using a photocentimeter. Now, here's how we're going to standardize the plants. So we already mentioned that one of the control variables is the length of the plant. How are we going to standardize the length of the plant, right? So a little bit of that. So, well, rather than how we're going to lengthen the plant, I mean, how we're going to fix the length, we're standardizing the plants by making it a constant length. How do we produce the two different light intensities? By moving the lamp closer and farther, using the sodium hydrogen carbonate, which is going to be which is going to provide carbon dioxide, allowing the plant to acclimatize for 5 minutes before you start the timer. and then you place the photocentimeter and you start the timer and you start collection for 5 minutes after the plant is acclimate. How do you pronounce that word? I sure hope I don't get distracted as I search what this word means. Acclimatize. The hell? Acclimatize. That sounds stupid. Acclimatize. Acclimatize. Okay, listen. So, okay, and is a climate a word? Sorry, I didn't close that. A climate. Yes, and you pronounce this by saying acclimate. I don't. Okay, sure. Acclimatize. Acclimate. So, yeah. Now this is all given there in the method that we learned over here. Leave for 5 minutes, and then we're gonna set up the bench lamp, and we're gonna be setting the photocentimeter and leaving for 5 minutes and recording the amount of uh, gas that's collected. Right. So 5 minutes for acclimatization, another 5 minutes for gas collection. Okay. Then one variable needs to be controlled in its method of control. So now that's the okay. So now remember how we're going to be changing some factors for that. So we already know that light intensity is what we're going to be doing over here, right? So therefore we're going to have to put the other things controlled. So temperature, CO2 content in the water, what else? Light wavelength, right? So those things have to be temperature, wavelength of light, pH, and how, right? So temperature, we're going to be using a thermostatic water bath, or was it thermostatic control water bath or something? So we can control the wavelength of light by using a filter. What is a TC water bath? A TC temperature control water bath is a specialized laboratory instrument designed for heating or cooling samples to a specific constant temperature for a long period of time. Sucks. Okay. So there you go. There you have it. Temperature or wavelength of light. Obviously, we didn't really bring up CO2, but if I want to bring that up, then maybe I could have said like controlling CO2 content in the water by using like a specific amount or a controlled amount. Oh, of sodium hydrogen carbonate, half a teaspoon or half a spatula. Okay, then okay. No, this is important. This is for the analysis section. So we want to repeat the experiment to give mean and SD. standard deviation, method of calculating rate. So both of these things are pretty important. Okay, so how would we calculate rate? Method of calculating rate. So volume of gas collected over time, and repeat the experiment to give mean and standard deviation. Okay, so let's just repeat what we learned so far. So that is going to be, okay, so dependent, okay, so dv plus how to measure it, then um, how to control, okay, so like method of control variable plant, okay, then okay, then turn on lamp, um, and collect gas for 5 minutes. So that'll be a point, that'll be a point, wait, yeah, that'll be a point. Then I think we're done, and then we just do repeat experiment and calculate mean plus SD, and what is it? Calculate rate by volume divided by time. One variable that needs to be controlled. So, okay. Control temp with TC water bath. Control light wavelength with filter. Okay. Okay, now this is another pretty important detail. How your results should be recorded, how it should be recorded, presented, and analyzed in order to draw conclusions for your investigation. So they're not asking for the conclusions, or maybe they are, but they're asking for how it should, the method of recording, the method of presenting, and the method of analysis in order to get those conclusions. Now, we already are aware of all the times we have a graph and we're supposed to conclude something from the graph. This time, we're going to be making that graph, right? That we're going to be making that data. So how would we record, how would we present, and how would we analyze the data to make a conclusion? Okay, so it's pretty simple. 3-marker question. You take raw data with headings and units. So this time around, okay, yeah. So first you have a table for your raw data, and okay, here they're asking you to place in headings and units with means calculated from repeats. Okay, so this is all pretty standard because we asked to calculate to do repeats and calculate the means already, and also add in headings. So that's also important. And I'm pretty sure they make this word really important that you're supposed to use the raw data. So initially, to record your data, you're going to be placing the table. Then to present the data, you're going to be using a bar graph format with labeled axes. And then t-test, I would personally not use a bar graph. I would use a scatter graph, right? So I would use a scatter graph format and always label your axes, and then use a correlation test difference, okay, with line graph showing more than 2 different line intensities. So you can then use the name of that test, the Spearman's rank test, or Spearman's rank correlation test. Notice that when you have a bar graph, you're using the t-test with the bar graph showing only 2 different light intensities, and then when you're using a scatter graph, you're using a correlation test. Okay. Now we're supposed to suggest 2 limitations and then to propose 2, but okay, now 2 ways to fix it is not there. Suggest 2 limitations of your method. So let's see if we can already guess them. So this is not performed in the natural habitat of the results might not be completely accurate as we're not growing the plant in its natural habitat, as well as the fact that the plant does not represent all aquatic plants, right? So that's also another thing. So here they say that it's difficult to control temperature even though we're using a TCA water bath. Idea that actual growing conditions in the river change during the day. So, okay, fine. So the fact that in real life temperature, biotic condition, abiotic conditions, stuff like that aren't constant. So yeah, it is not like a very natural representation. It might not be a fair representation. It's difficult to measure small volumes of gas with precision. So you can say some of the oxygen may dissolve in the water, right? Or may not be collected. So it's difficult to measure those small amounts of O2. Don't say amounts, say volumes. Volumes is important. And then difficult to control the surface area of the leaves. Now, that is to say that even if we have the same lengths, it's difficult to have because one of the control variables needs to be that these leaves have to be the same. Now, this is difficult because it's hard to control the surface area of these 2 leaves, but it is easy to control the length of these 2 leaves. In the same way, another control variable is the temperature, and it's hard to control the temperature and keep it fixed at a certain thing. So that's another problem. So think about the limitations as in, what are some limitations of collecting the DV? What are some limitations of controlling the CV? And what are some limitations of the results that we might find? Why wouldn't they be as accurate, or why aren't they as representative of real life, right? So for DV, it would be that it's difficult to measure all the gas that's produced, all the oxygen O2 that is released, because some may get dissolved and some may not get collected. CV, it's hard to it is hard to control the surface area of the plant and the temperature of the water bath or the beaker. And R is the results may not be as accurate because in real life growing conditions inside the river, abiotic conditions like temperature change spontaneously throughout the day. As for the results, how they should be recorded is going to be raw data is going to be recorded on a table with headings and as well as with calculated means from the repeats. It should be presented on a scatter graph with axes labeled, and it should be analyzed by using a Spearman's correlation test. Okay, boom. 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 So now let's do October 2024. Ain't nothing like a holiday. Tell me why ain't nothing like. Oh shit, so this is there as well. Okay, more or less it should be the exact same. But yeah, I could definitely see the T values coming in this. So I need to be careful. So mitosis occurs in growing roots. So a scientist allowed pea seeds to start growing at a concentration of oxygen. Pea seeds were grown in oxygen concentration of 5 and 20%. After 2 days, the root tips were removed. And the tips were stained and observed under a microscope. The mitotic index of the... Okay, this is not actually what we're talking about. This is measuring the mitotic index. Is that one of the core practicals? Core practicals... IEL Bio. Tell me why ain't nothing but it. Tell me why ain't Tell me why I never wanna hear you say I want it. Growth of microorganisms, temperature and the development of organisms, respiration in yeast affected different antibiotics. Stages of mitosis, observe and draw upon structures, tensile strength, antimicrobial properties. Okay. I guess growth of microorganisms is mitotic index. Nope. Tell me why. Okay. Should I care about this? Can't tell. কি হইতেছে ভাই ভাল লাগে না Okay, fine. So they don't ask you to calculate T. There should be a question that asks you to calculate T here. And so then you can see that T is more than the critical value. So therefore, we have to reject the null hypothesis and just explain what that is. You have to say that it's significant. We can also say here, give this value, but I don't think this is important. It's a 3 marker. Hold on. Well, how to extend the investigation. Okay, wait, so what is it? Grown under 20 have a larger mitotic index than those grown on. You can see probability is 0.05%. I mean, oh, it's unlikeliness, right? See that it's higher in 9 than it is as you go lower down. How do you tell that? Describe how the scientists could extend this investigation to collect more data. Use different temperatures, use different oxygen concentrations, use different times, use different plant species. So this could also be another thing that we could do in our own experiments. How would we extend the investigation to collect more data to either support or reject the hypothesis? So we would use different control variables, so different temperatures, different plant species, different times of day, or different times like measured. Then also using different oxygen concentrations. So in this case, it would be carbon dioxide concentrations, right? Because that's also another thing. So changing another control variable. Now we have the pondweed. Now this plant grows in ponds and rivers throughout North America. Student reads that pondweed grows fastest near the water surface. Wait, hold on. Is this a difficult one? Okay, this looks a bit more difficult. How many core practicals are there for 11? Quite a few. Okay. 10, 11, 12. I don't know if I have enough time. So first, the preliminary practical work. This is easy. We don't even need to read the question. The further away it is, the lower the rate of photosynthesis. So preliminary practical work. So what would that be? So we can imagine suitable way of measuring ROP, or like suitable way of changing Okay, so suitable length of pond weed, suitable temperature for photosynthesis, suitable method of gas production, measuring gas production, suitable depth of water or light intensity, suitable method of changing. So I said suitable way of changing light intensity. Okay. So some other things I could have said is other control variables. So instead of suitable length, talk about suitable temp, pH, and then it's and not only changing suitable way of changing the light density, but also suitable. Well, suitable light intensity makes no sense because we're changing it. So I don't understand that. It's a bit weird. So I'm fine with not putting that as a point because it's not a it's not a control variable. So it's not like it's not the same way as the last 3 points or last these 2 points, right? Okay, now devise a detailed method how you could control and monitor important variables. So it's the same exact thing. Okay, so in beaker place. Sorry, first thing is measure gas. O2 gas produced. Photocentimeter. দরজা বন্ধ করো তো Okay, measure O2 gas produced using photocentimeter. Think about the control variables. Think about the other things. Okay, so think about how we're going to method of controlling features of the plant, right? So like standardizing plants. So sure plants are all same length. Ensure temperature of water is controlled using TC water bath. Ensure okay. Place half a table, half a insert half a spatula of sodium hydrogen carbonate. Let plant acclimatize for 5 minutes. God, that was such a memories. I love school memories. I love school memories. Especially I love all the school memories where stupid memories. Hate school. Hate school. Okay. What else? I mean, just repeat experiment and calculate mean and SD and Sure, yeah. Calculate rate by vol divided by time. Okay, I think that's fine. So let's just so here we go. Measure O2 gas produced using photocentimeter. So O2 gas released per unit time. Okay, I didn't write that. Photocentimeter method is standardizing plants, controlling the length. Two different light intensities. Yeah. Hydrogen carbonate. What? Oh no, did I not say hydrogen carbonate? I did say hydrogen carbonate. Plan to acclimatize. 5 minutes for this. Temperature. Okay. Calculating rate. Repeat whole around. Calculating MSD. Boom. That's pretty much every point. Okay. How to record, present, and analyze. So, record raw data on a table with headings and means calculated from the repeat experiments. Present data on a scatter graph with axis labeled and oh my god, excuse me, with axis labeled and perform a correlation test using experiment rank correlation test or something, right? So table for raw data with headings and units with the means calculated from repeats. Scatter graph with labeled axis, use a correlation test. So two limitations. So limitations for, remember, CV, DV, and R. So dependent variables, why can't we control it? Because some of the O2 produced may dissolve, and it's hard to collect small amounts of carbon dioxide produced. Control variable, it's hard to control the temperature of the water, it's hard to control the surface area of the plant. And results, the results may not be completely accurate because in nature the abiotic conditions like temperature change throughout the day. So controlled surface area, the problem with apparatus, oxygen dissolves in the water. Yeah. So I'll always mention surface area and okay, leaves of plant. I'll always mention surface area and oxygen being dissolved. That's for sure. Okay, that's it. Now so we're done with these two. Do we do these now, or do we go into core practical 11? And I guess we can just gloss through these other ones. They are question ones, so those should be fine. Okay, so let me just open them up. We're gonna have to open the mark scheme too. That's annoying. Okay, so June 2020 question 3. No, let's do 21 and 22 question 1. 21 and 22 question 1. January, October 21. So that's gonna be paper 6. and all of the lights down, 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 down. Question 3. No, question 1, he said. Okay, there you go. Okay, just give me 1 second. Should be crazy. Should be crazy. Crazy, crazy, crazy business. It's crazy business. Okay. Oh, increase size a bit. What about now? 16. Oh my god. Okay. So, what is it gonna be? So, dependent variable is what you produced, measured using photo centimeter. Okay, wait. Is it synthometer, centimeter? What is it actually? It is photosyntho. So synthometer. Synthometer. Okay, what else? Oh, I notice how we don't even talk about the foil, by the way. Maybe in this case we will. Insert blah. Leave plant to acclimatize for 5. Collect gas for 5. Repeat experiment and calc SD and mean and calc mean and SD. Calc rate using vol over time. Okay. So use at least 2 different colored filters. Sodium, hydrogen, carbonate added. Bond rate to acclimatize. So I don't know necessarily if we wrote this or this, but we did write this, we did write this, we did write this. Identification control of one variable, temperature using heat shield. I mean, TC water bath should be fine. The repeat calculate means, so that's 5 marks right there. Okay. Oh, this is interesting. So explain how features of the grana and the chloroplast enable photosynthesis to occur. So this is beyond the topic. So so features in the grana. What the hell is the grana? Grana is multiple grana. The chloroplast enable photosynthesis to occur. So you have high surface area of the photosystem, or high high so for surface area of the chlorophyll pigment. Um, um, Enzymes present in the what's it called? I don't even know what it's called. In the cytoplasm is the matrix. To perform Calvin cycle. Um, Electron transport proteins in the membrane for electron transport chain. ATP synthase present in the membrane. Structural membrane, large surface area for photosystems. Pigments to absorb light. Enzymes for light dependent reactions. Electron carriers. Okay. Then October 2022. So that's it. Okay. So Grana. Maybe you should study a little bit of the chapter itself, but like I am so lazy. Done temperature on the rate of photosynthesis this time. So again, it's the same thing. How do we change the temperature using a TC water bath? Keep what control variables the same. Keep the light intensity the same. Keep the light, keep the this thingy the same. The light wavelength the same using a filter or using white light. So using a photocentimeter to measure volume of gas. Use of at least 5 appropriate stated temperatures. Okay, yeah, okay. Use different temperatures or 5 appropriate temperatures. Leave time for bond beta acclimatized, sodium, hydrogen, carbon added in excess, volume of gas collected in 5 minutes. Identification control of one variable, so we will control light intensity by keeping them at a set distance, or using a wavelength using the lamps or filters. The pH is always buffer, that's always the easiest one. Method calculating rate, which is volumes divided by time. Then describe how, so this is always the second question that joins alongside it. Some question regarding it, like regarding the syllabus, more like a unit 4 topic, right? So describe how oxygen is produced during photosynthesis. So how is it produced? it's produced it's produced through that thing, right? That other thing in the light independent reactions. No, the light dependent reactions. Where not the electrons. Is it? I actually don't remember, but I do remember it's in the light-dependent reactions. And it in order to produce any D plus, or no, in order to produce H plus, also releases oxygen or something. So photolysis, that's the thing. So water splits to form oxygen by photolysis. Electrons removed from water. Two oxygen molecules combine to form one. Yeah. So simply put, you can just say water splits to form oxygen by photolysis. And then two oxygen molecules, two oxygen atoms combine to form one oxygen molecule. This is going to be it's going to be a oxidation. So H2 split water split to form oxygen by photolysis by removing the electrons. And so then two oxygen molecules combine to form water. Okay. Next we have let's just check October 23 and 2024 because I'm pretty sure there's probably that secondary question that. So October 23. October 24. So this is going to be question number 4. Question number 4. Okay, it is 9 marks. There probably wouldn't be a second part to that. Yeah, okay. Honestly, paper 6 is easy. I don't know what this announced guy was talking about. Okay, fine. Well, to be fair, there's a lot of stuff, so it's fine. Okay, and we'll do 2024 October. I'm actually now done. So, what the? So now let's read through Core Practical 11, but let's give ourselves like a 7-minute break. Okay, I'll be back. Type. Don't joke that. No. Joke. My eye bags feel so heavy, yet I can't seem to guide. Spontaneity. Why hasn't it grown into your heart yet? I am not who you think I am. Illusion of contentment and doom. Well, I don't fear for me and you poverty, but I fear the world would be abundant with gifts for us as it has been for those before us, so that we would compete for them as they have competed for it, so it destroys us as it has destroyed them. So true. Let's watch the previous stream. There was one. This org synth one was bad. Like I literally just did not do anything. I did not read the chat on any of these. It's funny. Or I can't even read the chat if there was any. Oops. I want to see if when he came into the room. সময় নষ্ট করিস না
ফাইজান, নানা কথা বলতেছে কে? নানা গিয়ে করতেছে বলছে কেন কল করতেছে?
That's me. Camera on करते मैंने. It's me.
How did it turn on?
It was always on. I record while I study. Like it's recording right now, actually. It's recording me watching this. When did Jihan buy up here? Oh. Does the problem still happen when I no, it's fine. But it cuts off immediately, that's annoying.
Oh, dude, is it like the pitch thing? Like, if you scream really loudly, it'll cut off.
Where's Jihan buy up? Fuck your body.
I don't know. আমার না চোখ ছোট ছোট
Oh hell no, we got only 1 hour 30 minutes. Uh-oh. Uh-oh.
চুলকাতে লাগলো। এক্ষুনি ওষুধ নিয়ে আসাতে হবে আমাকে। Might be viral. I think বলে তো একদম ছোঁয়াচে সবার হবে। I think pink eye is viral. Yeah, it ছোঁয়াচে also contagious. এখন হঠাৎ করে কাজ করে একদম চুলকাতে লাগলো।
Oh, it's over. It's Jover. Eva like a beaver.
দুটার মধ্যে যদি নিয়ে চলে আসো একটু
শেখার জন্য
শো অফ করার জন্য, আর তাহলে হবে না। ওটা তোমাকে নিতে হবে যে তুমি তোমার শিক্ষাটাকে আরো স্ট্রং করার জন্য।
তুমি কি মনে করো যে ওই যে কালকে দাদি বলছিল নামাজ পড়ছিল কত তাহলে বিকজ ওই একটু শখ করতে চাইছিল নাকি ওই যে দিনের সময় বলছে না পড়া হয়নি না
এগুলো অনেক বেশি সিরিয়াসলি নেয় তোমার দাদি, ওটাই আমি চাচ্ছিলাম যে এত সিরিয়াস সব কিছু কত রাকাত নফল সুন্নতি যেটুকুই নামাজ পড়ি সেটুকুই তো মন দিয়ে ডিপলি পড়ি না, হয়তো তোমার দাদি পড়ে আমি অনেক মানুষ অনেক সুন্দর করে ইবাদত করে। আজকে উস্তাদ বলছিল যে যখন আমাদের সঙ্গে কেউ কোনো কথার রেসপন্স কোনো খেয়াল না করে অন্য অ্যাটেনশন মানে যতটুকু অ্যাটেনশন দেওয়া দরকার ততটুকু অ্যাটেনশন না দিয়ে কেউ যখন রেসপন্স করে তখন আমাদের কেমন লাগে? ভালো লাগে না, ওই রেসপন্সটাতে আমরা স্যাটিসফাইড হই না, তাই না? আল্লাহকে যখন আমরা রেসপন্ড করি, আল্লাহর সঙ্গে যখন কমিউনিকেট করি প্রেয়ার, আমাদের প্রেয়ারটা কেমন হওয়া উচিত? ফুল অ্যাটেনশন, কেয়ার, রেসপেক্ট, লাভ, obedience, সব কিছু শো করে নামাজটা তার মানে হওয়া উচিত, physically, mentally, verbally, or not only prayer, any kind of deed, like you're doing charity but you're not thinking of Allah, you're trying to show thinking of yourself inside, I'm doing something good, I feel proud of myself. But what if people do that because they're trying to like grow confidence? Like let's say ওরা অনেক weak, ওরা অনেক কি বলবো সবসময় scared যে মানুষ কি বলবে কি করবে, তাহলে ওটা কেমন হওয়া উচিত যে আলহামদুলিল্লাহ আল্লাহ তুমি আমাকে এই ভালো কাজটা করার সুযোগ দিয়েছো, এরকম you gave me a chance to do good, alhamdulillah. Okay, but like I still like alhamdulillah I did it, but I did it only because of you, that kind of thing, is that okay? Okay. Because some people sometimes they need to hear that they did something good, like or else they will always think that they want to hear from someone else. No, I mean they just want to know that they did something good. Some people, I mean I think that's human nature to want to hear that. If it is the nature, then it is not a good nature. It is not. I mean, some people they think that they've accomplished nothing in their life until they they realize that they did something good, and that's the kind of thing, right? মানুষের উচিত আরেকজন মানুষকে ওয়েল তুমি যেটা বলছো ওটাও ঠিক আছে, হ্যাঁ তুমি কোন রকম এনভায়রনমেন্টে আছো দেখ, আসলে পড়ো ডিসকাশন করো, এসিও দিবি না কেন দিবি না এসি দাও কেন জোরে দিলে তো হয়ে যাবে, হালকা ইলেকট্রিসিটি করার দরকার কি? তোমার পরীক্ষা শুরু হচ্ছে আর 1 আওয়ার, ফাতেমার 1 আওয়ার। ভাই আমার পড়তে হবে, তাড়াতাড়ি করতে হবে। Ecology of habitat. Okay, studying okay, using quadrats and transects to determine the distribution and abundance of organisms. Okay, yeah. So distribution and abundance, very important two things. We always try to find out the distribution and abundance of organisms, and we can use quadrats and transects to do those things. And then we can also measure the abiotic factors, which we already know more or less. Abiotic factors which are appropriate to this habitat. I've made videos on abiotic factors. You better go and watch that shit. Okay. If you don't know what that is. I don't really remember it either. It's okay. Aladinic. Okay. Measuring of abiotic factors appropriate to the habitat. Okay. So, holy yab. Oh my god, I ain't reading a lot. Just kidding. It's probably the ones with like the t-test and shit. But I just opened like a question, just to see, just to see. Jan 23, question 4. Jan 23. Nitrate ion concentration is an abiotic factor, so I'm pretty sure that's what we're talking about. Who is this guy talking to? Opsons. Wow. So this looks a lot more like what we're learning. Not the initial bit, because for Jan 23 they did mention that there is question 1 that's REB5, then there's core practical 9, 11, and 13 in this question. So I wish they just wrote the question number, man. So annoying. So various abiotic factors that affect the abundance of and the distribution of a certain species can be investigated. So light intensity, distance from a large street, river, lake path, river depth, and the area itself, the habitat itself. Okay, so these abiotic factors will affect the distribution. Well, I think there is definitely more. There's like pH, temperature, water content, etc., etc., etc. But okay, sure. Equipment list is we need to have a quadrat, we need to have a transect. Quadrat is the one with the grid, 0.25 meters squared. The transect is the one that goes in one direction. That's going to be the non-random distribution or something. That's going to be 20-meter belt. We have a clipboard with a pen, an identification book, and measuring equipment depending on the factor being investigated. For instance, a light sensor for measuring light intensity. So the abiotic factor being investigated. If we're going to be measuring an abiotic factor, then we need to have a light sensor, or we need to have some kind of measuring equipment depending on that. So remember, the abiotic factors that we can measure is light intensity, distance from river depth, and area. And what those abiotic factors are appropriate to each habitat. So here's the method, one with quadrats and one with transects. So when carrying out an ecological study of a habitat, there are two methods of sampling. Okay, sampling is you take a small area as a representation of the whole area. There are two methods of sampling. One is using transects and one is using quadrats. Okay? So what is the method of what we're doing? We're doing an ecological study of a habitat is known as sampling. So there are two methods to this. One is a transect, which samples across an area and measures an environmental gradient, whereas this is two environmentally different areas. This samples two environmentally different areas. So the method one is quadrats, where we're going to be sampling two environmentally different areas. We draw a large grid over the images or maps of 2 over 3 areas being sampled at, assign a numerical scale to the grid, and create a coordinate grid. So we have an image or map of 2 out of 3 areas being sampled. 2 out of 3 is to make it even more random, right? So we have 3 images. Out of 3 of them, you take 2 and you draw a large grid over them, assigning a numerical scale to the grid or labeling the axes and creating a coordinate grid. Then we take a random number generator to generate 20 coordinates for the first area, which is where the quadrats will be placed. Placing the quadrat at the generated coordinates and counting the number of the species being investigated in the quadrat or percentage cover. So this is important. We place the quadrats at the so we make the grid, label the grid, use number generator to generate 20 coordinates for the first area, and then we're going to be placing the quadrats at those coordinates and counting the number of species being investigated in the quadrat, aka the percentage cover. If a box is more than half filled, then count that as a full box. If the box of the quadrat is more than half filled by the species being investigated, count that as a full box, and then calculate the percentage of full boxes counted, or full organisms, I guess. If investigating small species or species that are hard to distinguish from each other, then percentage cover is the most practical way to measure abundance, whereas if investigating a species like daisies that are easy to count, then record species frequency. Record the abundance in a suitable table. So these are less than half, so we don't count these. But these are more than half, so we will count this. Now, when it is a species that is easy to identify like daisies, then we will record the species frequency. But if it's a species that's hard to distinguish from each other, or it's hard to even notice, then percentage cover is the most practical way to measure this abundance. And we can do it by, again, counting the number of species being investigated. Repeat sub 3 with the remaining 19 quadrat samples and repeats 2 to 5 in the other sampling areas. So that's the entire process for quadrat. I just want to quickly confirm what they mean by this. Oh, damn. Okay. Aladinic. What is the difference between between percentage cover and species frequency when doing a sampling experiment using Quadrat. In ecological sampling, the choice between percentage cover and species frequency depends on the morphology of the species and how feasible it is to distinguish individuals. So, species frequency measures how often a species appears across your sample set. It's an all or nothing metric for each quadrat. By definition, it is the probability of finding a species within any single quadrat. It's the number of quadrats. Take species divided by the total number of quadrats times 100. Easy for distinct, easy to count individuals like daisies or snails. Pros or cons is it's very fast to record but provides no information on the density or the bulk of the species once it is found. I guess, but isn't that a problem with like quadrats in general? Okay. Percentage cover measures the proportion of the ground area occupied by the vertical projection of the species. So a definition is estimate of the area covered by the species within a single quadrat. So this doesn't talk about the whole area, this talks about the single quadrat. No, it also talks about the single quadrat. Okay. Okay, so it tells us the probability of finding it, but the probability of finding it doesn't necessarily mean how much of it covers the area, right? It could be like you find one, but it's very, very common that you'll find one. Whereas the percentage cover will tell us how many we'll find. So if there's 200, you could assume that I'll very often find 200, right? Calculation is usually done by counting how many small square grids within the quadrat are more than half filled by the organism. When to use essential for grasses, mosses, or spreading plants, and individual roots are hard to identify. Protocols gives a better representation of the biomass and the dominance of the species in the ecosystem, though it can be more subjective and time-consuming. Okay, buddy. What time is it? 11. 11. Alright, so for the transect, we will choose an area with a clear environmental gradient and the abiotic factor. Abiotic factor is a much more important thing here because we want to find the environmental gradient. Environmental gradient means the environment, the difference in the non-living, right, environment. So clear environmental gradient, the abiotic factor being investigated, for instance, under a tree where it is shaded out in the open where there is no shade. So you can see that there's a clear abiotic factor being investigated. The transect is more often for the abiotic factor, whereas quadrat is more for the species or the organism. Lay the belt across the gradient, starting in the shaded area. Place the quadrat at the 0 meter mark and count the number of species being investigated in the quadrat. Place the quadrat at the 0 meter mark. So first you lay the belt across the gradient starting from the shaded area. And then you place the quadrat at the 0 and count the number of species being investigated in the quadrat, or percentage cover. So since we're talking about percentage cover, then that means it's like over half of it has to be inside the thing for it to count. So you count the number, aka the percentage cover, and record the results in a suitable table. Also take a measurement of the independent variable being investigated. So in this case, since we're measuring light intensity abiotic factor, we're going to record it using a light intensity sensor. So then we repeat step 2, but placing the quadrant every 2 meters down the transect. So that means we will have taken 10 samples across the 20-meter belt. And then we're going to repeat steps 1 3 in different areas of the site with the same environmental gradient being investigated. This means that you end up with multiple results for each distance. Remember I was drinking juice during the experiment? No, nothing. So here we have what we would look like. So area, let's say we had 3 areas of which we took 2. So we would have 20 samples in area 1, 20 samples in area 2, and this should be area 2. And we will have the percentage cover, or the frequency. So the percentage cover would be for, again, undistinguishable creatures, where frequency would be for distinguishable. Frequency is the transects that we're in divided by the total number of transects, so divided by 20. Percentage cover is just simply how many they were in a single thing. Actually, wait. What's the frequency again? Number of quadrats contained species divided by total number of quadrats sampled. So for species frequency, if I did say 20 samples, no, if I did 20 quadrats, I would have to use I would have to divide by 20. So therefore all the quadrats will have same species frequency. How would I calculate species frequency for a single quadrat? Species frequency is a value calculated for the entire site, not a single. You're correct that if you sample 20, your denominator is 20. However, the numerator changes based on how many of the quadrats actually contain the species. If I find 5, then the frequency is 25. You get one single percentage that represents the whole area that you studied. Yeah, if 5 of my quadrats found the daisies out of 20, then the frequency is 25%. Okay, B. I'm yawning so much, man. Aladinic. Here we have area 1, area 2, and area 3, where we're going to place the 20-meter belt, and from meter 0 to every 2 meters, we're going to be placing a quadrat and calculating the species, the percentage cover, as well as measuring the abiotic factor using whatever. So if it's light intensity, we would be measuring using a light sensor. And so you can see 0, 2, 4, 6 all the way to 20. So in total you'll have 10. Area 1, Area 2, and Area 3. So we have light intensity, which is the abiotic factor. And then the second thing that we're measuring is percentage cover frequency. So now here we go. Analysis is after the thing is collected. Remember, these are raw data with your headings and your units, and your means from your repeat experiments. After the experiment and data collection is completed, then obviously you're going to present the data on a graph or something like that. But analysis-wise, you're going to carry out a statistical test to determine whether your results have you obtained are significant. This is essentially, in other words, you're rejecting or accepting a null hypothesis. Transect experiment is using a Spearman's rank correlation coefficient to determine whether there's a correlation between the independent and dependent variable and whether or not the correlation is significant. Quadrat experiment is after the experiment, anomalous results and calculate a mean percentage cover for frequency. After the experiment, exclude anomalous results and calculate a mean percentage cover or frequency for each area. These can be statistically compared using the student's t-test, which determines whether or not there is a significant difference between the means. Allogeneic. I feel great, alhamdulillah. I walk really clear, my mind work and still that's the goal. When I saw in that room, I realized this person is an individual that deserves respect, time, and care. Which is I try to give whatever I give, and I let fear, now I respect. I feel like we have realized it's best to know. Mesh is getting distracted. However, that's happening in your equilibrium. Stop, it's not worth it. Okay, but there's a journal to Yes, about it really good. Well, I was really apologize for you. I was really confused and turned back. And I thought that really anything wrong with that picture. The experience that you just didn't know was true for that real just to see if I'm not. I think people can talk about this and I'm doing a bit of working, trying to jump in. One of the things to say you said, you try to keep efficient rather than have a stream. Great. You're what really talking about something that you're actually crazy over. Oh, this is so cute. Why did I keep talking to her? I don't know. Should have left the hole. Should have left her, bro. What are you doing? What are you doing? God, man. Freaking out so much. Poor kid. Shit. Which day was that? I wanna know. 12/11? Oh my god. Made my first dish on 13/11. So that is November 13th, yeah. I'm crazy on you. I'm crazy on you. Nice connection with Quran today. Good work too. Good vibes. Good vibes. I did finish 6B. The 20-minute method works, alhamdulillah. You need to think about how to stay away from using AI for research. You need to go through the dots, read spec. No problem if no blender. Yeah. Talk about life. This wasn't for 21st of November. Honestly, I think I should journal pivotal moments of my life more often. Yes, I don't read them, but when I do, that moment sinks back into me and feels so crazy. Let's see if it does. Although most of the day is mundane, those little moments is what makes the whole day a game changer. So journal. Yesterday would have been so good for it, and you have a crap ton of empty space in this book. Might as well just use it on my own. As of right now, I hope to better myself and figure out what's best for me, because I know things look nice, and I hope they truly are what I think they are, but they probably won't be, and that's the truth I'm preparing for. Alongside, question mark. Here he goes again, lol. Just show up, don't give up, show up. I told her a lot, dot, and we talked afterwards, dot, bleh. Yeah, okay, I'm never reading this book again. Oi! Oh. Okay, after the experiment and data collection, you carry out a statistical test. Blah, blah, blah. Yep. So just Lamayo, just use the Spearman's test, Lamayo. T-test, Spearman's test. Yeah. For the transect, use Spearman's rank correlation coefficient, because you have two data types, and for quadrant you only have one, which is mean percentage cover. So you're going to use students' t-test to determine whether or not there's a difference between two or more areas, significant difference between two or more areas. Oh, I hate studying. Give me money, give me money. Where the money? Where's the money? Sorry. Where the money?
What is this Kapoor? What are we thinking? This give me that money, give me that money. Hey, 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 hey. With the money, yeah. With the money, yeah. What? With the money. Ah. Where is the money? Give me the money. Type sheet. Type sheet. Type sheet. Type. Type. Don't even joke loud. Wow. Money, money. Give me, give me, give me, give me, give me the money. Give me, give me the money. Give me the, give me the, give me the money. Jan 2025 had 2 questions in a row about this shit. That's crazy. Okay. Jan 23, June 23. Cholo. Jan 23, QP, boom. MS, boom. Gasser Sula. Gasser Sula. Give me Sula. Wait, cholo. What am I doing? Okay. Question 4. Sula. So, grassless flightless grasshopper, grassless flighthopper, Brachyspis robustus.
Okay. My fault, David Bernora. This is important. Copyright on David Banana. Okay. This endangered species is only found in one small part of New Zealand. Grasshoppers. Man, I'm so stupid. The grasshoppers are approximately 4 centimeters long. The grasshoppers are most active. I don't give a damn. Hypothesis. So here we have, what's the independent variable? Gravel road and natural gravel area. Unused gravel road and natural gravel. So the population on the natural gravel will be larger than the population on the unused gravel. Ho ho! Okay. Big statements, huh? Plan investigation to do the thing. So describe preliminary work. So remember, the suitable stuff. So suitable this, suitable that, suitable this, suitable that. You know. So um, so my guess is gonna be like suitable transect belt length, suitable quadrat size, suitable time of day to perform experiment, suitable area to perform experiment. What else? Suitable. What about control variables? Calculating this thing, percentage cover or frequency. That's all I got. That's all me got. Okay, so suitable method of identifying the species. Okay, so that's another thing, right? Because we're identifying a species now. So suitable way of actually getting to know, like identifying the species. What does the species look like? How do we know it's a suitable method to stimulate grasshopper movement because they're camouflaged? Okay, so this is why I should have read the question. Find the time of day or month, or the best time of day or month to do the experiment. Suitable time of day to perform experiment, to do study or whatever. Find a suitable sampling counting method. Suitable methods transact size of quadrat, capture and release nuts. Okay. Suitable weather conditions. So I got money. I got money. Okay. Excuse me. Quadrat size, time of day. That's it. So identifiable. How to identify the species. How to identify best weather to do it, the experiment, and also suitable way to stimulate grasshopper movement because they're camouflaged. Okay, so you can't really see them, but if you have a way to stimulate them to move, then we can actually see them. That's the whole thing. Whoa. I didn't mean to do that. Okay, so not this. Not this. So again, Since it's hard to identify the species because they might look like others, we need suitable way to identify the species, suitable weather, suitable time of day, and what's the last one? A suitable way to stimulate them to move because they're camouflaged and we won't be able to count them or see them otherwise. Devise a detailed method on how you control and monitor the important variables. So let's just look at the answer first because this is our first time looking through these. Um, so what's the dependent variable? It's going to be the dependent variable. Dependent variable is the number of, like, technically the thing that's changing, or the independent variable is the environment, right? So the dependent variable is going to be the number of grasshoppers per unit area. Okay, that's the other thing that we need to make sure to be right. Is it going to be the rate of photosynthesis, or sorry, it's the oxygen produced over time. That's the dependent variable. Okay. Then method of producing standardized sample sites. Standardized counting on both sites allow use of quadrats. Two variables to be monitored. So that's going to be the type of soil or type of ground, as well as show the light intensity. Okay, no. So these are control variables. So we're okay. No, to be controlled. Okay. So that's gonna be temperature, time of day, pH, light intensity. Yeah. Describe how one variable can be controlled or monitored. So light sensor for light intensity. Repeats for one or neither sample area, or thermometer for temperature. Describe how one okay, the repeats for one sample area, and then repeat experiments at different times of year and calculate the population density. So shit, bro, we got 30 minutes. Oh shit. We got one more paper, two more paper, three. Shit. Gotta lock in. This is 8 marks. This should be the last bit, right? Oh no, there's gonna be two more. There's this, and then there's the limitations. Okay, fine. So, oh man, I'm a bit worried. Who? Yuri Master. Lane point. Wasting time! I'm proud of the shit! Meow 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 meow. Okay, lock in. Lock the frickin' the flip. So when we talking about this, when we talking about that, talking about this, talking about that. So that's gonna be, so first of all, dependent variable is the number of grasshoppers per unit area. Then we talk about controlling shit. So control the size of sample sites using fixed quadrats, same size quadrats. Um, Standard method on both sides. So it's gonna be uh So notice how we're first describing the dependent variable as always, then controls. Control size of the sample sites using fixed quadrats, control temperature and light intensity using thermometer and light sensor. And then after that, it's pretty much the experiment. Repeat experiment for one sample area, calculate mean. Repeat experiment for one sample area, calculate mean. Repeat whole experiment for another time of the year, calculate bulk density. This is it. Now, how do we do this? So record all raw data. Record raw data on a table with headings and units and means from the repeat experiments. Present the data on a bar graph with the axis labeled and analyze the data using this is going to be Spearman's correlation coefficient. Draw a table with headings and appropriate units with means calculated from repeats. Bar graph with it and uses statistical test for difference. The difference if it's significant. Then limitations. So remember, dependent variable, control variable, and result. So result should be fine. Maybe actually no. Maybe we're scaring off the insect, so we're disturbing it. It's not going to be the same if it was completely natural. The dependent variable would be okay. So dependent variable is the hard to count, hard to distinguish the hard to distinguish the grasshopper, hard to see the grasshopper, or not all grasshoppers will be counted. Control variable is hard to control temperature and light intensity and weather. And results is we might scare away, or we might scare away the grasshoppers, so the recordings or the data might not be accurate for what it is naturally. Okay, so difficulty in identifying, difficulty in seeing, difficulty in ensuring grasshopper counted as a new individual. I did not say this. This is a pretty big one because, like, is it fitting in the quadrant or not, right? Difficult to measure or monitor light intensity sampled once, so population may change over time. Okay, I did not write that. So I did this, I did this, I did this. I did not do this or this. So this is gonna be, this is trying to say that this is trying to say excuse me, this is trying to say that having difficulty in checking whether or not more than half is going to be filled inside the quadrant box. This is trying to say that what date is it? Tuesday. Say that the population will change over time because we only need to sample it once. We need to sample it multiple times a year. Yeah, hard to distinguish the grasshopper, hard to see the grasshopper, hard to monitor and control variables, and hard to judge whether more than half of the grasshoppers inside the box. Oh, okay, this is like, what if it moves? You don't know if it's the same one or not. Okay. Control variable is, no. The results is you only sampled once, so results may change in another time of the year, or population may change over time. Okay, so that's another thing. Okay, boom. Now we're looking at June 2023, question number 2. So this is question number 2. Scientists observed that this was often near a species of tree of this. Cactus plants often grow next to trees. 100 locations were selected around and 10 quadrat was placed at each location. Presence of absence of each species were recorded. So we're looking at the thingy. We're looking at not percentage cover but the frequency. Species frequency. It suggests one possible benefit to cactus plants of growing near mimosa trees. Um, they'll be shaded by the tree, uh, which helps them decrease temperature so they can conserve water. So like less water is evaporated. Then so there's one risk carrying out the investigation. Thorns on the cactus, so they might get hurt. So you have to wear protective gear like wearing gloves and also being careful around the cactus. Um, Shelter from wind, sun, so reduce water loss. Okay. Spines, appropriate description. Allergic reaction, so wearing like a mask and keeping a distance, or letting the teacher know that you have a thing, but this is scientist. Okay. So now we have, okay, this is a whole thing. Shit. Okay, that's all though. So, okay, the 2025 one is type paper. Wait, is it Jan 25 or what's it again? Okay, since we're already here, let's just complete this. So, yeah, so we can see the results. Out of 100, you can see that 60 were present. Okay, mimosa being present and cactus being present. So over here, we can see that if mimosa were present, 60 were present of cactus, 4 were absent. If mimosa was absent, you have 16 being present of cactus and 20 being present of cactus. So you can see the highest is over here and over here. An odds ratio can be determined to check if the presence of mimosa trees has any effect on the presence of cactus. Calculate an odds ratio using the following steps. Give your answer to two significant figures. So both are present divided by both are absent, and present absent oh no, this is okay, I got it. So both are present divided by absent present. So okay, and then present absent and both are absent. Present absent. Both are absent. Okay, 18.75. So 18.75 is more than 1, so it's so since it's much more greater than 1, it has a very high likelihood. Presence of mimosa trees has a very high likelihood cactus plants being present. 18.75. Okay. Now abiotic factors in the soil cannot be controlled. Yes, however, these variables can be measured to confirm that these plants are growing in similar conditions. Say to abiotic in the soil, that could be okay. So pH using like a universal indicator, and okay, that could affect it. So we have pH and we have, let's say, could temperature of the soil be one? It could, right? Um, maybe even like pH, water content, or like pH and temperature. And choose one of the variables, so pH. State the effect it could have on the results if the measure were not similar. No, the pH was different, then the results would have been smaller. Like results. It would have been altered. It would have been different. Temperature, pH, water content. The results are not valid. Description of expected result. Description of effect must be directional, e.g. that is increase in temperature, increase in enzyme activity. Of the results, on the results if the values are not similar. Wait, what? Okay, I don't think I read the question. These plants are growing in similar conditions. If they are not growing in similar conditions, then yeah. Okay. Okay, and then this question, what's it? Jan 25. One sec. Jan 25, Oct 25. No, it'll be like 30, 40 minutes. Oh, this is gonna take ages. I don't think I can do this in time. Oh, why? Why would they do something like this? This is an awful idea. So BioJan 2025. Question number 2. Okay, I'm gonna close some stuff. Cause I got too much stuff open right now. I got too much stuff open right now. So here's an actual question. Damn. Okay. Okay, B. Describe a sampling method that could be used to collect valid data. So near an acacia tree. So this is again going to be a different, like this is going to be away from a tree. So we're gonna have to use a transect. Okay. So dependent variable is going to be the number of termites. Measuring the dependent variable is using quadrats, using a fixed size of a quadrat. I don't know, monitoring and controlling the temperature and light intensity. Um, what else? Not sure. So the rounding sampling to select trees, measure distances from trees and termite mounds, compare trees that are close to mounds and not close to mounds, decide if a tree is fruiting. Oh god. As soon observe that the trees only produce fruit when they grew next to a termite mound. Oh dear. Well, okay, I'm gonna stop streaming here, but yeah, there you have it. 3 hours of nothing, more or less.